Hello viewers, in this video I'll be discussing about bile pigments, and to introduce myself, I am Mahesh, working as an assistant professor in Department of Pharmaceutical Chemistry, College of Pharmaceutical Sciences, Dayanand Sagar University, Bengaluru. At the end of this session you will be able to understand what are bile pigments, how these bile pigments are formed in the body, the fate of bile pigments, what is hyperbilirubinemia, what are porphyrins, and what are porphyrias. Bile pigments. They are the excretory products of heme metabolism. Heme is found in blood, and these pigments called bilirubin and biliverdin are intensely colored. Bilirubin can be seen in the skin during jaundice, and biliverdin, which is green in color, can be seen during bruishes. Heme is most important for porphyrin containing compound. Towards the end of this presentation, I'll be discussing what this porphyrin means. These heme, it is primarily synthesized in the liver and the erythrocyte producing cells of bone marrow. Succinyl coenzyme A with glycine forms delta amino levulinate, which in turn forms heme. We'll be discussing about the heme production in the further slides. Pigments are being formed from heme, and this is the structure of heme, which in the presence of the enzyme heme oxygenase gets converted to biliverdin. Biliverdin in the presence of enzyme biliverdin reductase gets converted to bilirubin, and this takes place in the blood. We look into what happens to the heme or these bile pigments after it is formed in the blood. Here it comes in the blood, hemoglobin. From hemoglobin we get heme in the presence of globulin, and in the presence of heme oxygenase enzyme, this heme is getting converted to biliverdin. Biliverdin reductase converts biliverdin to bilirubin. So this much we have seen in the previous slide, and over here these bilirubin, which is being synthesized in the blood, it goes to the liver through blood. The bilirubin or the water soluble bilirubin in the liver in the presence of UDP glucuronic acid gets converted to bilirubin diglucuronide. Even this bilirubin diglucuronide is water soluble. This step takes place in the liver, and this bilirubin diglucuronide via bile duct enters into the intestine, and in intestine, in the presence of the intestinal bacteria, it forms urobilinogen, and this urobilinogen gets converted to stercobilin, which is being excreted in through feces. So stercobilin over here is responsible for the color or the brown coloration of the feces. Urobilinogen also gets reabsorbed into the blood, which passes through the kidney, and through kidney it is getting converted to urobilin, and in through urobilin it is through excreted in urine. And the color of the urine, the yellow coloration of urine, is due to the urobilin content in the urine. So this is the fate of the bile pigments. This is another illustration of the bilirubin transport or the formation and the fate of bilirubin, which has been explained in the previous two slides. When the bilirubin is not being utilized properly by the liver or by the kidney, what happens over here is like there is accumulation of this bilirubin in the blood. So this accumulation of bilirubin in the blood leads to hyperbilirubinemia, the interference at one or more processes of bilirubin utilization or process of excretion of this bilirubin leads to a condition known as hyperbilirubinemia, meaning it is characterized by abnormally elevated levels of bilirubin in blood. Hyperbilirubinemia include the yellowish or yellowing of the skin, sclera of eyes, and mucous membranes in the conditions like jaundice. It is detectable when the total plasma bilirubin levels exceeds 2 milligrams per 100 ml. So in the example what I have given you, the picture what I have given you, you can see the yellowish or yellow coloring of the sclera of the eyes over here. Causes of this hyperbilirubinemia include increased bilirubin production, reduced bilirubin uptake by liver, disrupted intracellular conjugation, generally in the neonatal jaundice, hepatitis, cirrhosis, impaired liver function cases. So when this hyperbilirubinemia, it leads to increase in free bilirubin which causes hemolysis. Also, disrupted secretion of bilirubin into bile canaliculi or intra or extra hepatic bile duct obstruction re- results in the rise in conjugated bilirubin levels, which also results in hyperbilirubinemia. For these hyperbilirubinemia includes phototherapy, administration of phenobarbital, albumin ingestion or infusion as 25%, and clofibrate. As mentioned earlier, the structure of heme contains porphyrins. So the porphyrins are cyclic compounds composed of four pyrrole ring held together by methynyl groups or methynyl bridges. Metal ions bind with nitrogen atoms of pyrrole rings to form complexes. Heme is an iron containing porphyrin, while chlorophyll is magnesium containing porphyrin. There are two types of porphyrins. Type 1 porphyrin, when the substituent group on the 8 positions are symmetrically arranged, they are called as type 1 porphyrins. Example is uroporphyrin 1. And type 3 porphyrin, they contain asymmetric group at the 8 positions and are more common in biological porphyrias. These are the metabolic disorders of heme synthesis characterized by increased excretion of porphyrins or porphyrin precursors. There are two types of porphyrias, namely erythropoietic porphyria, where enzyme deficiency occurs in the erythrocytes, and hepatic porphyria, where enzyme defect lies in the liver. Session. I hope you are able to understand what are bile pigments, the formation of bile pigments, fate of bile pigments, what is hyperbilirubinemia, what are porphyrins, and what are porphyria. Thank you very much. Namaste.